Let's talk now about the shape and structure of protein. Shape is critical to the function of a protein. A protein's shape depends on 4 levels of structure: primary, secondary, tertiary, and quaternary. The primary structure of a protein is the sequence of amino acids that comprise it. Each protein consists of a unique sequence. So maybe we have alanine, leucine, serine, lysine, or valine, leucine, leucine, alanine, or lysine, alanine, serine, lysine, or you get the idea, any of the infinite combinations of amino acids we can come up with to make a protein. Changes in the primary structure of a protein are changes in its amino acid sequence. Changing an amino acid in a protein changes its primary structure and can affect its overall structure and ability to function. One example of this is sickle cell disease. Sickle cell disease is an example of a single amino acid defect. So one change in an amino acid makes a normal red blood cell look like the sickled red blood cell and function differently. Sickle cell disease is a blood disorder specifically involving hemoglobin, which carries oxygen in the blood. A single glutamate amino acid is replaced in the primary sequence by valine. So I have this one substitution. The result changes the overall shape of the hemoglobin molecule and does not allow it to properly carry oxygen. Secondary structure is a result of hydrogen bond formation between amine and carboxyl groups of amino acids in each polypeptide chain. Depending on where the groups are relative to one another, the secondary structure takes the shape of an alpha helix or a pleated sheet. So you can see in the diagram at right, we have some hydrogen bonds shown here and here and here, and those are hydrogen bonds between neighboring amino acids. Okay. The R groups, the amino acids themselves, do not play a role in secondary structure because every amino acid has an amino group and a carboxyl group. Here's some visual examples. In orange, you can see pretty clearly the helix, that's your alpha helix, and these pleated sheets are what we see in green here, these sheets of amino acids folded onto one another. Tertiary structure is the overall 3D shape of the polypeptide. It results from the clustering of hydrophobic and hydrophilic R groups and bonds between them along the helices and pleats. You can see an example of tertiary structure shown. The function of a protein is determined by its shape, its tertiary structure. Its shape is driven by chemistry, but it is the shape, not the chemistry, that dictates the function. Each sequence of amino acids folds in a different way, as each amino acid in the chain interacts with water and the other amino acids in the protein uniquely. For instance, upon contacting water, a protein can fold into grooves that function as binding sites for other molecules. Changes in heat, pH, and salinity can cause proteins to unfold and lose their functionality. This is known as denaturation. In the fried egg example shown at right, the egg's protein has undergone denaturation and loss of solubility caused by the high rise in the temperature of the egg during the cooking process. So we see that denaturation as the formation of the solid egg white.